So environmental variability, of course, is at the core of a lot of biology, at least ecology. I mean, it's the sort of foundation concept of adaptation, you know, that led Darwin looking at the sort of different food sources and Darwin's finches, and then the concept of adaptation, that the particular individuals within a population that do better under certain circumstances, pass their genes on at a greater rate, so allele frequencies change over time in populations, leading to speciation. And that's sort of the classic concept that still underlies a lot of the ecology and evolution in particular. But in the last sort of, well, I mean, the concepts are not that new, but it really has become a dominant, or is becoming a dominant concept now. On top of adaptation, you have epigenetic changes, which means there are changes to the gene expression programs that do not alter the nucleotide sequence of the DNA itself. So the DNA molecule stays the same, but you have little groups put on that sort of switch on and off genes. And that's really a much more effective, or sort of transpiring to be a much more effective way for organisms to change their phenotype in response to external stimuli. It's very rapid, much more rapid than genetic adaptation, which by its very definition takes many generations, whereas epigenetic processes can only take 2 or 3 weeks, or even days. So it's sort of this instantaneous, almost relative to adaptation response to an environment. And of course the dominant thought is that that's beneficial, because what the organism can do, it can adjust its phenotype to prevailing conditions and then avoid potentially negative impacts of the environment. So there is this sort of almost hope in climate change, for example, that plasticity will increase the resilience of animals to changing conditions, including climate change and heat waves and whatever else comes with it. And then, of course, again, you have that response would then, if it is beneficial, it would determine what organisms you find where and how many of them, so biogeography, biodiversity. So what I'll talk about, I'll start with definition of plasticity. Even though it's a very well established concept in biology, there is no agreement necessarily of what it actually is. And I'll sort of step away a little bit from the complexity definition we heard in the last session and provide a more sort of functional approach. And then I'll talk a bit more about what sort of environmental information do animals need to actually change the phenotype. These are sort of outstanding questions, if you like. There is no resolution to that yet. And I will point out where I think the really interesting questions lie. And then I'll finish with a short discussion. Is it actually beneficial? You know, is plasticity always beneficial? Can we make that assumption? And is there evidence against it? So if you look at a simple reaction rate, chemical reaction rate, so I mean, everyone who's done first year chemistry or high school chemistry will know this, right? You have your reagents in a test tube. You want to speed up the reaction, you hold it over a Bunsen burner. You put more energy into that system in here, and you get this sort of increase in reaction rate with an increase in temperature. Now, if you put an animal into a warmer environment and measure some sort of a rate function, what I have here is this locomotive performance. So performance is an evolutionary concept. It is any trait an animal has that is related to fitness, to its reproductive success, but it's not a measure of reproductive success. So what happens is exactly the same thing. You put an animal into a warmer environment and the reaction rate will increase. In biological systems it will decrease again because there's sort of a damaging level. So membranes break down, enzymes lose their structure, so there's a decrease again. This is called a thermal performance curve. It's one of those currencies, I think, in ecology and evolution. So far so good. These two are the same. So we have increases in reaction rate. So what we see here is a thermodynamic constraint, right? There's not enough energy down here. You put more energy in the system, it increases. Just thermodynamics. So I would not call that plasticity in my view. It's a constraint, but not a response by the animal to its environment. So what then is plasticity? And that's what, as Esmee said, leads me to crocodiles. And that's how I started my career. That's what my PhD was on. And for some years afterwards, I worked on it. And we put different transmitters into these animals, sort of implanted things in their body cavity to measure internal body temperature. We measured cardiovascular parameters, all from a distance while watching the animals. So I spent a lot of time measuring physiological responses of the animal, watch what they do at different times of the year. So I got them to know very intimately. And that sort of led to my interest in plasticity. Because what I realized is that they do very different things at different times of year. Now, if we go back to the graph I've shown you before, this is a lab study on crocodiles, and we measured, we exposed the animal to summer conditions in the lab, so radiation levels, temperatures were all sort of set given what we measured in the field in summer. And then we simply throw the animals at different acute test temperatures and measure their swimming performance, and we get this sort of typical thermal performance curve. The really interesting thing is now, if you throw the same animal into winter conditions, you get this, right? You get a shift in the whole curve. So in this summer curve, the animals thermoregulate to the sort of high 20s, and it coincides with their maximal performance. You put the animal in winter conditions, that whole curve shifts, and maximum performance then coincides with the regulated body temperature. And I would call that plasticity, because it's a regulated response by the animal in response to environmental change. And in this case, it probably is beneficial because it doesn't lose out on any performance despite a sort of 10-degree difference in its thermal environment. So it's one of the very few clear-cut examples, and that's partly why I showed it here, in the literature. Most of the time, the data are much more messy. So just to summarize, there's a regulated response, there's a shift in the whole curve, right? Whereas the change here is just a thermodynamic constraint. And I think that's an important point to realize. And in this case, they maintain performance, so they do compensate for the potentially negative effects of, in my example here, a decrease in temperature. Or it can be an increase, of course, everything is relative. So phenotypic plasticity then depends on epigenetics. So what I was talking about, so yeah, I saw the light, then soon after I worked on crocodiles, and now I just work on small fish where I get hundreds in about half an hour rather than having the logistic nightmare of working with crocodiles. It's the same thing. I mean, most organisms show some level of plasticity. So it's not specific to any taxon. So what I talked about is acclimation within adult organisms. So even adults can change their physiological responses. But of course, the early environment experienced by the very early embryos, so the blastogastrula, the very, very early stages, will determine the phenotype of the animal, and it will determine how plastic the adults will be. And the very early embryonic stages, their phenotype or their responses will be determined by the environment the gametes experience in their parents. And then that goes back several generations to grandparents, great-grandparents. I think the literature has some good data showing that that can go back 3 to 5 generations. So if your grandmothers smoked while they were pregnant, you may well have less plasticity down there. So there is that inheritance that you mentioned as well, that it's not changes in the nucleotide, it's not genetic adaptation, it's simply regulating, putting marks on the molecule that change the phenotype, or change the gene expression rather than the genes themselves. Now all of these things work together. We'll ignore all of these because I'll keep it simple and work together to then cause this sort of stylized here response where you have a different curve for different environments. So these are the chronic environments, and these thermal performance curves shift backwards and forwards. And the upshot is that performance doesn't change. If the animal was fixed and there was no plasticity, what you would get is this. The curve stays the same, and that shift in environment translates to a difference in performance. So what that would mean, the animal in winter is much slower, or you can again have it on this side of the curve, that the animal in summer would be damaged by hot temperatures. So that's the crux of plasticity, and that's the crux of thinking it is beneficial because there's this constant performance despite of what the environment does. It's very powerful, of course, so if that really were the case, then animals would be very resilient to things like climate change. So regulated response, there's reduced variance in fitness-related traits, so compare these two to this one here, and it's compensation for environmental effects. So if you look at the identity of the individual, so let's stick with our crocodile here. So the individual is comprised of different states, and each state is defined by a function. So it's a function-valued trait. It's not a point, it's not a single value. The whole trait is represented by this sort of what's essentially a Gaussian function. And the questions that then sort of arise is what mechanisms underlie this state shift between, in our case, different thermal environments? What's the environmental signal? What are the regulatory processes? And I sort of borrow a little bit of the terminology of information theory. I'm not an expert in information theory, but it really lends itself well to think about plasticity and how animals are tied to their environment. So you can think of an organism at its time, at its current state, let's say, as a Bayesian body. It has a certain amount of priors, so the current state is defined by its priors. And what it does all the time is send out, sense the environment continuously. So if there's a predictable environmental change, like the sun gets up in the morning and it heats up and cools down again as a normal sort of dial pattern, there's no news here, right? That's already there, right? It happens with great certainty. So there's no need to update the priors. But what we get, for example, if we get an unpredictable variation, such as a heat wave, let's say, it could also be translated to winter and summer. But what I plotted here is sort of a heat wave over a few days. That's unpredictable, right? So the animal cannot predict that. And that requires an updating of the priors. Because what happens now is that these priors are not, there's a difference between the internal and external state. So you have this Bayesian updating of priors with new information. And that's the crux, the information. So Gregory Bateson defined that as the difference that makes a difference, so the famous social scientist. And that's exactly the crux that we don't know. What is the difference that makes a difference? By how much does the environment have to change to cause this updating of the priors? One of the really important questions. If you think of climate change, you may get a really low rate of change over a long period of time, but that rate is too low to make a difference, but the animal may get damaged over time. So if we knew exactly what the information is we need, then we would also know how resilient animals may be in the face of change. So if you sort of summarize this, the animal goes from one state to the next, which is identified by these performance curves. That's my definition of plasticity, and what lies in between them is information. So information is commonly expressed as minus log probability of an event occurring, and I already mentioned that. So the sun gets up in the morning, probabilities are very high, very little information in that. For the more sort of unexpected events that occur with low probability, what happens? There's a lot of information in that for the animal to update its priors. So that's sort of a way of thinking about how animals are tied to their environment. So information content increases with uncertainty, and predictable events carry little information. Again, it's all sort of meant in a Bayesian sense. And then, of course, the question again, what rates and magnitudes of change? And temperature, for example, can provide enough information to cause that switch between states in an animal. And now the next question that sort of obviously leads on from there is, how's the environmental information processed? I mean, it's one thing to have an environment that changes and then you have a phenotype, but how are they connected? We know there's information somewhere, but that information somehow needs to be processed to lead to different phenotypes. And it's a little sort of cartoon I've put together here where I think is sort of a conceptual model of how this is all processed. So we have our environment here, our unpredictable change, that contains information. Again, I put that question mark there because we don't know exactly what that information is that can cause that change. That change is sensed by the nervous system. So there's particular proteins, transient receptor potential ion channels that can sense particular temperatures and send that information to the brain. And the brain, this is a crocodile brain, by the way, and the brain then translates that signal to endocrine information. So you expose an animal to different temperatures and you, for example, will have a very strong thyroid response. And thyroid receptors are nuclear receptors that then have the potential to change whole gene expression programs. So you have information in the environment, you have the sensing, you have temperature sensors, light sensors, all sorts of sensors that are connected to the brain, particularly hypothalamus. It translates it to changes in hormone levels, for example. It can also be receptor densities, receptor sensitivities. But here is another interesting point, because we have another level of information here, because we have an increase in hormone levels, but by how much does a hormone level have to change before it has a phenotypic effect? Again, an unknown entity. So you have two places where this information environment somehow is translated to endocrine information. But we have to know here again how much is enough. There may be that is sensed and passed on, but the change in hormone levels is not enough to cause a phenotypic change. So again, there is a really important, outstanding question there. And then hormones regulate epigenetics. Again, most of my work is on thyroid hormones. There's lots of thyroid response elements on the major proteins that mediate DNA methylation, for example, in the promoter region. So you have a mechanistic pathway, if you like, where this cascade can lead to epigenetic changes, or you can have direct changes, and that will change the phenotype. So in my mind, that's sort of actually I just wrote a ground proposal on that, so it's very close to my heart. But I think this is a really good conceptual pathway that can be tested in different ways. And our planes we have different transgenic fish we can use there and different pharmacological protocols to test this information and how much we need to get a phenotypic change. Now, I brushed over lots of things here just to get this sort of concept of information across, but let me go back to what I said at the beginning. Is plasticity always beneficial? Now, in an evolutionary sense, if plasticity was beneficial, you would think that plastic individuals would outcompete non-plastic individuals in a sort of classical Darwinian sense, because plastic individuals would have slightly more, produce slightly more offspring, so their genotype, if it is genetic somewhere. I should mention here to epigenetics, of course, also genetic, right? The two are not divorced, because all the proteins that mediate epigenetic changes have evolved. So there is not a fundamental separation between the two. They're a continuum, if you like. But we've done an experiment a while ago on mosquito fish, these little brown fish in Australia. You find them anywhere. They've been distributed around the world by, I think, the US military to eat mosquitoes. They ate everything else, of course. And as part of that, the interest was we sort of measured these individuals twice. So we exposed them to one temperature, measured the thermal performance curve, then exposed the same individual to a different temperature, measured the thermal performance curve, and then measure, and that allowed us to measure how plastic each individual was. And that's what we expressed in this acclimation capacity here for each individual. So if it's 1, we have this crocodile curve, if you like, perfect compensation for environmental change. If it's 0, then there's no compensation at all. And we use lots of wild animals, and what I've shown here is sort of 7 mesocosm studies, so 600-litre tanks where we put founder individuals in this case, found individuals with high acclimation capacities and some populations that low acclimation capacities. And over several months or a year, we let the population build up in a sort of naturalistic environment. And then we sampled the population again and determined acclimation capacity of the offspring after sort of 3 or 4 generations. And the really astounding thing is that there is no signal, because almost all populations, bar this one maybe, have the full range of phenotypes in there. They have fish in there that are not plastic at all, and they have fish in there that perfectly compensate. And that sort of is quite interesting, and it's quite contrary to the idea that plasticity is always beneficial. And we got lots of fish. We measured 600 fish all up from wild in these mesocosm populations. So it's a fairly robust data set. Now the reasons for this, maybe there's a cost of plasticity. So it's all well and good. Animals are really plastic, but there's a hidden cost. And what we found is sort of there's a statistically significant, but really in its effect size, quite a weak indication that plastic individuals have greater oxidative damage. So there is a cost involved in that. So oxidative damage, reactive oxygen species produce the mitochondria, react with membranes and proteins, break them down. So that's possible. It may be. In the literature, people say there may be an energetic cost of expressing different phenotypes. I don't think there's a good basis for that, because whether you produce a blue or red curve will cost you the same energy. There may be a trade-off. So it may be that, great, the curve shifts, but you may have a change in maximums. And that's what we find. Again, biologically not a very strong signal, but we found that the animals that are really plastic have a slightly lower maximum under warm conditions. And that may just mean in the hot summers these fish live in shallow waters, often in very hot conditions like in Australia. That the ones that are not plastic in summer just outcompete the ones that are plastic, because they do just that little bit better. I don't know if that's true. It's a plausible explanation that we have not tested. But if there's some such trade-off, we're only looking at one trait here. There may be, in the multitude of traits that are happening, there may be some that actually go down in plastic animals. And then lastly, there may be sort of a bet hedging strategy. In ecology, it's called a portfolio effect, like a portfolio in invest in a stock market. You don't just buy stocks from one company, you buy stocks from lots of companies, because if one goes down, it's more likely that some others stay up. And that sort of concept has been translated to ecology. It's sort of for meta-populations. You have a lot of populations of one species. They all have slightly different characteristics. And if there's a major change in the environment coming through, it's more likely that some of them will make it if there's a diversity. And you can sort of apply that same concept to individuals as well. It may just be, because of trade-offs and costs of plasticity, it may be in an evolutionary sense beneficial to have the broad range of individuals here. So that plasticity is not necessarily beneficial, that there's also benefit to be not plastic. And I think for the sort of broader discussion for this forum, I think that's a good thought to have. I think that, I think it's an assumption around that plasticity is actually a good thing. Maybe not always, or maybe there are sort of sometimes and sometimes not. And these data would indicate that. I don't have an answer. We actually never followed this up. But it's a super interesting question, I think. So let me just sort of summarize the main questions that I come up with in my own mind and with this little talk. So one of them, I think, is a central question. What's the environmental information needed to induce a phenotypic change? Is it the rate of change? Is it the magnitude of change? I'm just talking about temperature. Of course, you can apply that to any other environmental variable as well, but you've got to boil it down to something to make it manageable. Change in rates and magnitudes, and then the translation of environmental information to endocrine information. There may be thresholds. It may be that there needs to be a certain information content in the environment before you get any endocrine response, and that may not be linear, it may not be proportional. Again, not known, but a super important question, which then leads to what endocrine information induces phenotypic changes. And if you understand those points, or those sort of that sequence of questions, if you can answer them, you're in a much better position to have make predictions of what climate change and other environmental changes will actually do to populations of animals. And I think that, to my mind, that really is one of the important frontiers, if you like, to understand how animals respond to environmental change. And then, of course, which hormones, is it circulating levels, is it receptor distribution or densities? There's all these sort of sub-questions in there. And then the benefits and constraints. Maybe we don't want plastic animals at all. So, it's a question that comes up. If you think plastic is good, but there's major trade-offs under particular environmental circumstances, then it's not beneficial at all. And maybe that's why we're seeing such a broad range of phenotypes within populations. I don't know. These are questions that are interesting and worthwhile answering. And I'll leave it at that.
Thank you so much. Speaking of plastic animals, which one calls to you as...
I have to take the duck.
Okay.
I love ducks.
You love ducks. Thank you so much, Yaron, for your beautiful talk. We will go to a very different field now, but then afterwards, of course, try to draw bridges. So I all invite you to do that together. So I'm very happy and honored to introduce you to Danqing Li. Danqing is associate professor at the Department of Chemical Engineering and Chemistry at the University of Eindhoven. She has her own lab, the Human Interactive Materials Lab, and I'm very excited to see many cool videos about what you're doing there. Danqing is a material scientist, as you will learn more about, and in her work she attempts to fill the field between molecular sciences such as synthetic organic chemistry and the dimensions of the more macroscopic materials, including morphology and shape control. Thank you so much for being here, and the floor is yours.
Thank you for the nice introduction. As said, that we have a lab in Eindhoven called Human Interactive Materials, abbreviated as HIM. We tried to find her or she, didn't work. But our lab is not exclusive for only men, we have many girls actually. So what we are doing is we are creating materials. Actually it is polymers, so plastic. And the question we want to answer is that what do we do with the so-called responsive materials or plastic? They mix to soft robotics. How do we make the application? What is the need application of those materials? So I'm trying to answer those questions via this presentation. So in our group, we try to make soft materials, polymer plastics, and those are responsive. And we are thinking of a number of applications, which I also want to discuss and share with you, is that first of all, as you can see here, we can create dynamic surfaces. This surface we hope to interact with a human. For example, by touching this surface, what you see here is not animation. It is really experimental measurement. The surface keeps changing from one state to another state. And we hope this change could introduce touch sensation to humans. And then from there, we can bring this to virtual reality. Nowadays, virtual reality mainly addresses the optical or the vision or the audio. And touch sensation is largely missed in virtual reality if you compare with the daily life. So we want to introduce the interaction with the material to virtual reality. We're also trying to answer other questions. For example, people with prosthetic arms, quite often the neuron, the mind doesn't feel the arm. You cannot control very well because the disconnection from the neural signal to the arm. So we are hoping to use the material to generate signals to bridge the connection. And also we are working on the 5G communication. You will see a little bit later. Come to here. So this is we are from the chemical department, so it is natural that I show you a number of molecules, but don't be scared by those molecules, because the next slide you'll feel more comfortable. Those molecules are generally used in your liquid crystal display. 100% of liquid crystal display contains the molecule I showed before. So this is a nice way to start your research, because since ever since you show some application, we are from technical university, always application oriented. Then you show to people, they say, oh, must be expensive. You say, no, because that's used on display. You say how cheap the display is, so don't worry about the price. So this is nice thing. Besides that, you also already build up enormous amount of knowledge on these materials, because optical is really used, fine-tuned on your display. What you are doing is that you are not looking at the display application or optical application in this sense. We are going to mechanical application for this material. So why we specifically use these materials? Why not the other polymers? And the very interesting properties of these polymers is that it has anisotropic deformation of these materials. What does that mean? First of all, you should think of this material, the molecule, like a rod shape, and they can, if you bring them together, they are so-called very well aligned, so at high order state. Once you start to decrease the alignment, so it is less ordered as you see here, what happens is that contraction and expansion happen simultaneously on the same material. So if you measure the thermal expansion coefficient, what you will see here is that along perpendicular to the general molecular direction, you have the expansion going to the thermal expansion coefficient goes to positive, which means the material expands. That is your common knowledge. Every day have the material, you heat it up, it expands. But this material, when you measure the direction that's parallel to the molecular direction, what you will see is that the thermal expansion coefficient start to decrease, and to a certain point it becomes negative. This means that we have shrinkage on this molecule as well, so expansion and shrinkage. What you can play around this is that you start to manipulate how the molecules are organized. For example, in this case, this is the cross-section. You should imagine this is cross-section of the polymer thin film of 6 micrometers. On the top, the molecules are organized parallel, and they start to rotate till the bottom one start to be perpendicular to the substrate. What happened is that when you heat it up by decrease the order parameter, is that it start to bend because the top contract, the bottom expand. And this can happen just on one single film. Normally you need bilayer structure, 2 layers with different thermal expansion coefficient, but in this case you can just realize the bending just within one film. And of course, with that we can go to further. That, for example, we can make the bending response to the humidity. What you see here is the film close to the water bath. So the humidity is higher, the same bending, same molecule alignment. Can also make this to respond to the light. And then eventually, for example, you can make this material connect to a rotating wheel, and as the belt to not rotating wheel, the expansion and the shrinkage can eventually rotating this wheel to realize certain functions. Now, since this molecular architecture in height, you can figure out many ways of how you can manipulate the molecule structure, and then start to play around with different structures. Here you can go from flat to the cone shape, like you can show here. And for example, you can make a wheel out of that, and all different kinds of the deformation we can imagine. And one thing we find interesting is that since we can make the surface, the substrate from flat to cone-like, or the other way around, like you can see here. And we say, okay, this is like an octopus suction cup, and then the octopus suckers using the vacuum to pick up things. So what we do is that we mimic this, and we make a film, and you can see that we can just pick up the glass plate that hundreds times heavier than this polymer. This time we use light to trigger the response. and then we can further even connect it to the entire robotic arm. Here under the UV light, here what you see, it is the support and the parallel arm, vertical arm made by the same material. Under the light can pick up something, rotate, release, and go back to the initial state. Besides that, we can also make the arm to grab then a weight that's again much heavier than it is itself. And I said we can also work on the optical communication. What you see here is that we're using these materials to deform a piece of rubber, and then therefore we can change the depth of focus just using light or using heat in this material. Now, another thing is that the video you saw before looks very nice, but they're slow. They're in the order of seconds. Quite often you want the deformation to be faster and faster. So, but then we are restricted by the nature of soft material. When it is soft, the deformation is naturally very slow. So to solve that, we are in the perfect place to solve this, because what you see here is that in the Netherlands, everyone has the experience. You have the umbrella in a rainy, windy day, and your umbrella just snaps to go to another direction very fast when the energy is stored enough. So we think of it. Okay, then we inspired by this, we also start making snap-through system. So this is the initial state. Then when it gets enough energy, it's popping up. Quite often the video we show always accelerated 5 times or so, but in this case we slow down the video because this happened in 70 milliseconds. You can see that it's fast snaps through snap deformation, and we further use this force generated by snap through to throw a ball. Now, as I said, in our lab, actually we think the material, if you think of a robotic application, actually those soft material cannot compete with the traditional robots at all, because it is slow, it doesn't exert enough force. So what we are thinking, what is the advantage? What we believe, it is the interact with the human of the soft material. So one of the nicest way to interact with the human is actually a bunch of fibers you can touch, navigate your hand around the system. Inspired by this, we start with, this is again a molecule, we're making sticky molecule, and we draw fibers from the substrate. Here, because the fiber is transparent, not very easy to see, but on the glass substrate, we draw many fibers, then we start to shine with the light. The fibers now all rotate, bend, and always bend towards the direction of the light as sunflowers. Since we need the fiber to interact with human, what you saw here is that the light is exactly just on the body of the fiber, then you cannot interact with the fiber. So the next step is to let the fiber to deform under the bottom instead of the top. So what we're trying to do is to shrink the bottom of the fiber. By the shrinking at the bottom of the fiber, the top bend out accordingly. And in order to do this, we would like to use electricity, because if you think of any application, you prefer electricity because that can be directly integrated to the existing electronic device. With that, we designed the electric circuit board, not me, but we have some is trained as the chemist from his older education, but in this case, I need to turn him into an electrical engineer. So this is the trick. And then he designed the circuit board from the fibers on top, and what you can see, the results is that. So this is the fiber, then we trigger, actually electricity is embedded under this fiber. That's also why the fiber looks green, because the circuit board is green. And we start to shrink this part, and you can see that fiber bends accordingly in this case. Now, the next step, we have one fiber we triggered by press the button, electricity is on, and then the next, I said to some, besides you now become electrical engineering, I need to become a computer science, because we need to, I want to drive the fibers by create the program on the app that you can slide with your finger to any direction, and your fiber should follow the swiping direction, and eventually the application of this is that we're going to use this for remote control, like people wearing the fiber, and under the distance, people do something and you can feel the touch on your skin. Now, so far we are talking about that we want the fiber or the material interact with people to bring the touch sensation. And we was asking ourselves, who needs the touch sensation most importantly? That's the answer is blind people. So far, blind people, they don't have any access to the digital book. They can buy the audio, there are many audio, but by touch, they don't have. They still have the cupboard, the paper and punch, put the hole there and the protrusions they feel. But then, since we have iPad, for example, computer, you can continuously refresh your content there, but they don't have the access to any ebook for blind people that can continuously refresh the message. So what we are doing is that using our techniques, since we are able to create the surface, the protrusions from flat surface to the pre-designed corrugated surface, then we design the dots according to the Braille display or Braille dots. Then we can switch this and on and off and selectively switch for any pattern. Therefore, we can connect this to the electronic book and get the blind people access to the Braille book. And now at this stage, we are working with psychologists to bring this to the blind people to test. What surprises you, it is not about the resolution. You always think, okay, they need high resolution, but the fact is that blind people need to get used to touch the plastic. They always used to touch the paper, not the plastic. Now, so since we can making the surface to be triggered under the electrical field, you see the example of the Braille display, but we can also making some other more random surfaces on top of the electrodes. This is the measurement you can see under the holographic microscope that the surface, by applying electric field, can go from flat state to rather corrugated state. And again, as I said, we are from technical university, we always want to solve problem. One of the problem is this poor guy, actually from China, was cleaning the solar cells. Monday he started from here, Friday he ends up here. Finally, clean all the solar panels, but then next week he has to start all over again because of the dust. So we were thinking we should help this poor man to solve his problem. And therefore we think of, okay, can this dynamic surface continuously oscillate? Can that be used for self-cleaning? For that reason, we bring the sand that is around 100 micrometer in diameter, put on our surface. And in the lab, we quite often making the sample size of 3 by 3 centimeter. In this extreme case, the area covered by the white sand is only 1 centimeter by 1 centimeter. Then we start to apply electrical field. You can see the signal is on, and you see that within 1 second, 96% of the sand is repaired from the surfaces. And this, of course, caught the attention. For example, NASA came to us, contact to us, say, yeah, we do have the same problem that our Mars rover operating on the Mars suffer from sandstone upon one sandstone, and the solar panels covered with the sand. There is no power anymore. For that reason, we lost 3.5 Mars rover already. Can you use your coating, for example, coat it on the solar panels, and then use your vibration like you saw from the previous video, and the surface start to vibrate. Take, use this to clean the panel. Actually, we can not only actually in this case, not only the solar panels on the Mars rover we can clean, but if you can think of the mechanical component, the optical component, all can be covered and keep the entire system clean. We didn't say yes after even they approached us for 4 times. The reason is the laboratory is not really equipped for large-scale manufacturing. That's the engineering aspect to stop us. In the previous video, what you saw previously, we always start from a flat surface to a corrugated surface. But what can be also interesting, we think we start from corrugated surface already, but now we start to inverse the surface. This means, as you can see here, the top becomes the bottom under the trigger, and the bottom can go back to the top. And we play a trick. And initially we put on the top of this wave kind of structure with adhesive. And this is the adhesive, is the muscle protein used to attach to the rock. And then we put the surface with the adhesive, and the bottom there is non-adhesive. And then if we can measure this, is that at the top over here, that's facing towards the air, you have much larger force than the bottom because there is no adhesive. What we are doing here is that then our coating can start with an adhesive surface, and we switching, we use light, can be electricity or whatever you like. Then we go to non-adhesive state. Adhesive state, we use this to grab an object, in this case in water. And then we can transport the object to the location we like. Then we switch on the light to go to the non-adhesive state, and we can release the object. And we developed this to provide actually an alternative solution for the pick-and-place system in industry. Pick-and-place system in industry always using vacuum, but vacuum is really can damage many delicate components like optical device, semiconductor device. They're really looking for an alternative way for that reason. So we propose to switch the surface from stickiness to non-stickiness in this case. With that, I would like to end off this work here. I mainly talk about how we envision that polymer can interact with people, especially how we can change the coating surface properties. What I didn't touch upon is that we actually can also, like our human skin, we look into our human skin, we can also eject liquid from the bulk of the coating to the surface to release drugs and to release chemicals. But then I mainly focus on the topography, and I hope I convinced you that the coating can be used for the human to interact with outside world. Actually, eventually, I hope the skin can also apply to the real robot, and they can interact besides the electronic way in a different way to communicate. And with that, I would like to thank the students contribute to the content of this presentation. We don't often dress like this. We will have a special event. And then also I would like to acknowledge the funding source, governmental funding source, and the industry source. And of course, for your attention. Thank you very much.
Thank you so much. Yes, you may please also see or feel or sense which spirit animal calls to you. Please, how you maybe have not heard that announcement in the morning. So you can pick up. Yes, as a token of our appreciation. Thank you. Thank you so much for this really fascinating talk. It was so amazing, and can I just work in your lab? Otherwise, I will just play with clay later. May I please, invite both of you to join the panel discussion. How are we microphone-wise? Are we just?
We can speak.
Yeah, for the online audience.
That's better for Zoom.
Okay. Yes. Of course, I will be very happy to invite someone for the stage in a bit. I just would really like to go into some burning questions that we have as a team, considering plasticity and also applying plasticity in new fields. So you've seen, Frank, in your talk, you're using plasticity a lot. It's a very well-known concept, and Danqing, in your talk, it was mentioned less, and it's also a concept that's maybe less used in your field. Could you both maybe reflect on each other's presentations, like how do you relate these systems or organisms or things you create? I think things that I saw that have some resemblances are states, like the crocodiles have different states, your materials can change states, so this shift between states, sensing the environment. So maybe what did you pick up anything on plasticity that you might apply in your field?
I think for me the most important is to improve the performance, the quality of the material, the plasticity. It is already a plastic polymer, plastic material. We can further use this to introduce the materials to a longer lifetime, for example. And I mentioned that in the extreme case, like mass over, not only actually about the mass fabrication, but also under the extreme case, extreme environmental conditions, we may use the different materials.
Thank you. And how do you?
Yeah, I thought about it when I listened to you, and I tried to think, okay, where does my concept fit here? But I mean, you said a couple of things already. I think. I mean, performance is one, in addition to what you said. I mean, but there's also the potential for trade-offs as well, isn't there? You know, in terms of you make a material that in one state adheres to a disk, but the trade-off maybe it performs worse in low temperatures or whatever, you know, I'm just making this up. So that's very similar to what organisms may experience as well, I think. So and that's the sort of thing I picked up. And of course the environment inducing different states where you have the light and then letting go. So I mean there clearly is a parallel. I can't quite get my head around the sort of regulated response versus constraint, but I suppose all of your things are regulated, aren't they, because they're designed. So it's an interesting parallel, I think.
Yes. Maybe I have, of course, many other questions, but I'd like to invite people. Ananda, you're welcome to come to the stage or stay there. Prefer.
I'll stay here. My name is Amanda. I'm in the consciousness biz. I have a question. Maybe I'd like to think through plasticity in terms of what might be an antithetical concept based on what you've sort of brought forward. So maybe I can ask sort of a short version of the question and then give the biopolitical preamble. I'm still thinking about artificial skins as well. So. But I think mostly, I'm first maybe directing something at Frank, which is the question, what would be a dysregulated response at the level of the organism, but also maybe at the level of the collectivity, maybe the species, or at the level of the environment? So I'm thinking about, yes, the plastic crocodile, but what about the not so plastic crocodile, the recalcitrant crocodile? What would the name for that dysregulation be? Would it be, is its response toxic? Is it die-off? Is it hypoxicity, oxidative damage, I think I heard in there? So I guess I'm wondering what the status of plasticity is vis-a-vis, you know, like in terms of life and its thriving, but what would plasticity be vis-a-vis another opposite like extinction? And if that sort of registers within you know, sort of the paradigm of study that you're thinking about. I mean, I'm also thinking about plasticity in terms of like the discourse of plasticity. Like I've heard a few sort of versions about plasticity in terms of use, energy use, power use, tool use. And I'm wondering about, you know, when things aren't useful, how do they register in a plastic paradigm? I'm thinking about many questions to do with environmental justice and one example, the use of the Inuit people. The discourse of plasticity as a pretense to displace the Inuit people and say you'll adapt to these conditions way far north because you are descended from the Thule people who were there before a period of climate change. So here is, and I do believe you mentioned the US military.
In terms of mosquito fish.
Yeah, studying mosquitoes, I know, but also I sometimes wonder where these concepts and the research of these concepts, like are they bankrolled by the US military? Do they serve the US military? But I mean, not just the US military, obviously, but there's a lot of research that is invested in creating conditions and environments and actually studying what responses are. So my question is, you know, what are the measures of a failed plasticity? Like, what concept do we use to say, no, this is enough. This is, you know, this is, so I think you were also mentioning many economic concepts, like where is the site of by which maybe the organism says, I'm trading off or I'm bet hedging, you know, is that a consciousness that's in the environment? Is it a consciousness that's in the organism? But here still, it's an understanding of ecology by way of economy. So what is how do we decide, or who is deciding what is the cost? This is too much a cost. And so this, I'm sort of thinking through, okay, what are the, you know, how do we get at plasticity by way of these other concepts that are bearing down in other systems of thinking?
Thank you, Emma. Quite some thoughts.
Thanks.
This doesn't all fall on you, I have to say. I enjoyed thinking about this.
I think just in terms of biological systems and animals, I always study, people like me study. I don't think, excuse me, the lack of plasticity has any value attached to it. Quite the opposite. I mean, I think the way I ended up, it can actually be very valuable to not be plastic. So I don't think we attach any value to it. I mean, there's the prejudice around that plasticity is good. So I mean, a lot of people may be disappointed when they run an experiment and find that these animals are not plastic. But I mean, I would say, well, that's actually super interesting because it tells you not plastic is good. So that's how I would look at the biology of it.
What would you call an animal like that?
I would simply call it has a low acclimation capacity and there's numbers on it. It's a calculation, I say it's 0.2. I call it 0.2 and 0.8. You know, I wouldn't have terminology that in any way would have a value attached to it other than the actual measured value, I think. Now, the other part to your question, how does that translate to the US military? Well, or other such things, other such entities, socioeconomic system? I think it's a really interesting question, in particular because those arguments are used by governments and politicians, and I think there's the sort of argument or the sort of approach by lots of governments is that individual responses are important rather than sort of fair government responses. So there is room for that, I think. Now, personally, I haven't been exposed to that. But your argument with, you may as well live over there now because we need this land here, I think it's plausible. But I have no first-hand experience. But my feeling, of course, is it's a totally unacceptable way to use, otherwise neutral biology or data. But I can see that potentially happening.
Thank you. My contribution here is going to be in two parts, I think. Time sometimes matters, doesn't it? I mean, time of development and so on, when we discussed this a bit this morning. And the trade-off, therefore, might be something which is beneficial in an early stage of development may be at the cost of something which may be detrimental in later. This is certainly true, we know, for fetal development in mammals, and in particular, we know that that is true in humans, that in situations of nutritional deficits of various kinds, there's an adaptation of the developing fetus which will alter its metabolism and its growth strategy. And it is a strategic decision, if you like. I don't mean conscious decision, but it's a physiologically strategic decision. But what is known is that this reduces plasticity in later life. For example, the cardiovascular responsiveness is more restricted, so that humans that are born small tend to have greater incidence of mortality from cardiovascular diseases in their 50s and 60s and so on. So there's a sort of clearly a trade-off going on there, and very, very difficult to work out on an evolutionary sense of what that would be. But this comes to my other point, which is the sort of value judgment type of thing. It concerned me immensely, this question of whether you want to adopt a value to any particular aspect of biological functionality. And I wrote a paper for a chapter of a book which warned of the potential for there being prejudicial ideas about the fact that women should be held responsible in their diets for the consequences of that in terms of fetal adaptation and therefore the consequences in later life. It seems rather strange saying that, but it's actually, women get held responsible for all sorts of things in different societies. And so I don't think we should, certainly we should not have a value judgment, it seems to me, about an ethical judgment, a moral judgment about whether it's good, bad or indifferent to have enhanced flexibility or adaptability. But that's a different question, isn't it, when considering the issue of the role of adaptability in the physiological process, if you see what I mean. So it enhances the ability of the system to maintain its integrity when it's under certain degrees of stress. And maintaining some kind of adaptability of that could be a good thing. In other words, so, which is why not hardwiring it can be a useful thing. So just leaving it epigenetic can be a good thing. Yet there are circumstances where it might be a better judgment to, as it were, make it more permanent. We know that some epigenetic changes get incorporated into the genome. That's been true of I think the adaptation to hypoxia in high altitudes. So populations that live at high altitudes have adaptations. And when you look at different populations in different parts of the world, they've clearly adapted in slightly different ways. Sorry, it was rather long.
Thank you, Ray. Let's have an overview. Yes, Una.
Yeah.
And then we'll go to the other student.
So I want to refer to the part where we were talking about plasticity trade-offs. So if there's a sweet spot between being plastic enough and not plastic enough. So neither extreme is beneficial for an organism, but that doesn't mean the same applies to an ecosystem, right? So if some organism, let's say, I think interesting case study would be non-native or formally called invasive species, such as lionfish. So is this lionfish just at this sweet spot so that it makes it beneficial for itself in terms of how much it's plastic, how much it's not, to have a reproductive success in the ecosystem at the expense of others? Or is it the lack of acclimation capacity that you were mentioning? And then we want to circle back, like, do we really want organisms to view them as like in this type of competitive context? Or do we want to look at the ecosystem and rather discuss plasticity as a function of everything which is connected in an ecosystem rather than what's beneficial for one species or not? Of course, that's not what we're trying to do, but interesting tangent also, earlier talk by Igor and by Peter about plasticity being a function of the strength of connections between the nodes, but also strength of connections. So the stronger the connections, the harder is it for plasticity to occur. So like, go back to the lionfish. Is this lionfish, as an organism, does it have like a strong multiple connections so that it's difficult for the thresholds to be exceeded? So I don't know, is lionfish plastic, or are the others lacking plasticity? You don't have to answer this question. But
I'm happy
it's
to
kind
at
of-
least
it was-
comment on the question.
I struggled to formulate this, but I hope you understand and others what I'm trying to pinpoint here.
Thanks, Uma.
Look, thanks very much. That is a really good question, you know, and I'm totally guilty of sort of brushing over a lot of detail, but you of course have to see an individual within its population and a population within the species within an ecosystem. And what I've talked about ignored all that. But you're absolutely right, that an individual like a mosquito fish that's invasive species, it's really plastic, it does really well in an environment, causes a lot of damage to the ecosystem. And that's a dimension that's super complex to determine, but really worthwhile. So the closest I come to it, we have some artificial food web experiments at the moment where we have different organisms we expose to different conditions, and then we look at energy flow depending on how plastic the organisms are. Just to try to get to that question, do individual responses, do they harm or benefit the whole system? It's very difficult to do, but a very good question. The other thing I want to say about acclimation and plasticity, again, is sort of we have to get a bit more complex, and I think the greatest cost, I think, and sort of was something that you also said, in plasticity is a mismatch, right? It's a time factor that if an animal during development is induced, plasticity is induced, but the optimal environment changes, then it has a mismatch later in life and it does very poorly. But what happens is that if the animal is well able to acclimate, it can compensate for that. So there are interesting interactions between between developmental trajectories and also between generations. And if you could place that complexity into the complexity of ecosystems, that would be beautiful, but it would be a challenge. I think someone with the time you have on your hands, I think you can start that.
I was wondering maybe if there are reactions that also cross this bridge to the more chemical and material science realm. Who has a reaction? Julia?
Thank you. Yes, my name is Julia Krug. I'm from Wageningen University. So I have a question about kind of bridging the definition of plasticity between the different fields, because you mentioned that you would describe the first curve of just the swimming speed of a crocodile in water at different temperatures as just a, like a thermodynamic response. So I would say abiotic response, or in that sense. And then the regulated response from the organism as like the plasticity aspect. But with Danqing Liu's research, we saw that it's the design of the material, right? And then in combination with the abiotic input, let's say, which changes the plasticity of the structure, for instance, right? So in that sense, I wonder how transferable the definition is in a sense, and then what would be kind of the core of plasticity between the two fields. That would be kind of my thought, and then maybe also to the other disciplines, what would be kind of the distinction if you think about a community or society? What would be just an effect which you say, like you said, it's just a thermodynamic response, so maybe within a community it's just a response because of X, Y, Z, or that is like really the plasticity element here, let's say.
Thank you, Julia. Maybe Danqing, would you like to react to this?
Maybe I want to listen.
Okay.
So I will take Julia's comment one step forward, and I will direct it to you, Danqing. So if I look on your lecture, you were talking a lot about molding systems and how to change the properties. Now, what we have noticed in the last 2 years that we were working together is that plasticity as a term, and this connects to what Julia was talking about, is sometimes used in some field as a very important concept. For example, in phenotypic plasticity in biology, as we all saw in this morning, or in complex system or thing like that. But in chemistry, where plasticity originated in material science, you can say, in physics of material science, it is not a very core concept that people use. And so what I would like, if you can do, is try to reflect why is it, and why is it, and if we can say something from that about not only about the chemical level, but also, for example, for people like before that were talking about how do we take plasticity on the social level, which is also over there. Plasticity is still not very much discussed. So if you can try to bridge from your perspective to what Peter was doing before, and then all the way up, and to try to say what makes in one place that it works, that plasticity will become a core concept, in one place that it's not.
That's actually quite a lot to ask in a few minutes. As you said, indeed, I think plasticity is really not the core concept in chemistry. Actually, when I get the invitation for this seminar, whatever, symposium, was actually the first time these words came to me. As a chemist, we don't really discuss this. I guess in the social concept, I can understand you discuss this a lot. In the chemistry, maybe, I hope maybe also there is the idea of moving towards different materials, concept like organic materials, instead of only focus on the specific polymer type or plasticity, I think.
But do you think that it can add to your field in any way?
Add to my field, I think definitely, but this is really requires like coming to, for people like a chemist, come to maybe more type of this symposium and to interact with social science in a way could come to a little bit more aware of not only the terminology but also this concept.
So if you will have to pinpoint one thing that plasticity could contribute to material science or to smart materials or to chemistry, what will it be?
Give me a little bit time.
We will get back to you. Let's see, we're approaching lunchtime, and I'm sure food is also very important for maintaining plasticity and levels. So how many questions or remarks are there still? A lot. Okay, you will have to also continue during lunch. Maybe I'll go to Ginny.
Hello, thank you so much for your talks. I'm a psychology student at the UVA, and I am actually very interested in a part where you talk about the Bayesian body of an alligator. And I found it very interesting because within statistics you also differentiate between different kind of schools, like Bayesian statistics and the hypothesis testing, which is the frequentist one. And I think during your talk, you also talked about statistical significance and effect size, which I would then probably attribute more to the frequentist one, because you also before talked about hypothesis testing. Now my question is, how did you work with this Bayesian concept of the body of an alligator? It's more conceptual interest, and I hope it's also concise enough for you to answer.
We need a new type of microphone that just snaps into our bodies.
Yeah, good question. Look, I just borrowed, I'm aware of Bayesian analysis versus frequentist versus randomization tests, and I'm on the Bayesian randomization side. But I really, for the talk and for my thinking, I just borrowed the concept. I think it just lends itself so well to express, you know, the ideas rather than actually analysing the animal or the data. I'm sure you could actually. I mean, you gave me an idea now, because it would be possible to do that. That could be really interesting. But what I talked about today, I really just borrowed that concept, yeah. But I'll think more about it, I think.
Thank you so much. Well, that's great. Ideas are already emerging. Maybe we have time for one last. Yes, if you could.
Yeah, thank you very much. I think this is a bit more of a broader reflective on the sort of the question of what plasticity is and how it operates on these different levels. And my own sort of experience also reflecting on the talks before is sort of this question of time, because we have sort of these, the reaction to changes in the environment. Well, then you're already talking about the changes in the environment. Then you have sort of the question of the agency on the level of consciousness of people who are making decisions versus on the biological level of physiological changes. And then also in terms of like the question of the environment and the kitchen metaphor, that you have like what is the experience of time within that process of healing. So I'm very curious, and then also in terms of circular causality, if it still makes sense to have a linear conception of time that underlies it. So maybe just for broader discussions still to come, that I'm very curious in those questions.
Thank you so much for actually already making the bridge to our third panel on the perception of time after the break. I don't know if you very shortly would like to reflect on time or to just...
I can make a quick comment. I think that's a really good comment you made, and I think everything in the world is more or less transient. There's not very many static things, but we just simplify it to more or less static things. But I think the challenge is, what I talked about, information and trying to find out which information is actually important, that in its very nature is transient, right? Because you measure rates of change and so on and so forth. So it's sort of a small way to address an issue of time that occurs in multiple ways. Also the time when plasticity is induced in your previous generations or during development. So I think it's a really interesting area and a good comment.
Thanks. I just have 2 questions for you that you don't need to answer now, but you can come back to us later with the tablets or... the question to you is, how did the crocodiles change your epigenetics? Yes, and the question to you is, how did these human interactive material that you work with, how did they impact your own sense of identity? So these are just open questions. I think then now we will end this session. So much to think about, so many more questions that hopefully you can advance further during the lunch break, which will be outside. And we will be back here at